I'd like to request Dr. Sunita Agarwal ma'am, Program Coordinator, Star College Scheme, to welcome our eminent guest and audience. Ma'am, please.
Thank you, Dr. Rashi. Good afternoon, everyone. I, Dr. Sunita Agarwal, on the behalf of Department of Microbiology, Institute of Economics, welcome you all on the occasion of International Microorganisms Day, an initiative taken by Portuguese Society of Microbiology in 2017 to spread the awareness of microbes' importance in our life, and to mark the day when A.V. Leeuwenhoek reported to the Royal Society of London about his first description of microbes. I am pleased to welcome Professor A.M. Deshmukh, founder and president, Microbiologists Society of India, whose untiring efforts and unstinted support to promote microbiology is a source of inspiration to all of us. I welcome you, sir. I would also like to welcome our director, ma'am, Professor Geeta Trilok Kumar, who consistently motivate and support us in all our endeavors. Unfortunately, due to some other pressing commitments, she is not able to join us today. I also welcome Dr. A. Archana, President, Delhi Unit, MSI, for her ever-available support to all of us. It's my honor and privilege to welcome our eminent speakers, Professor Js Vedi, sir, former Professor, Department of Microbiology, University of Delhi, South Campus, and Professor Shilpi Sharma, Department of Biochemical Engineering and Biotechnology, IIT Delhi, who kindly agreed to spare their valuable time to enlighten us on the topics of interest. Challenges of addressing antimicrobial resistance and the unmatched power of microorganisms, an ode to the unseen heroes. I welcome you, sir. I welcome you, ma'am. Last but not the least, I welcome all my colleagues from different colleges and dear students. Before we begin with the talk by Professor Js Vedi, I would request Professor Deshmukh sir to address the participants. I welcome you, sir. Professor Deshmukh sir, please.
Thank you, madam. Really, I am very happy today on the occasion of International Microorganism Day. You are celebrating the day and updating the knowledge of the students on the very serious issue related to the microbiology. And the right person in India who can authentically talk on antimicrobial resistance, Dr. Virdi is here. I really appreciate the efforts of your college, your team, your director, who has tempted you to arrange such beautiful event in the microbiology. Of course, as you have said in your lecture, that tireless efforts of Dr. Deshmukh. I humbly reply to your statement that tireless efforts many microbiologists in India are taking. I am only collecting them. I am doing nothing. So I am inspiring them. I am trying to encourage them. And many research workers, talents, teachers, volunteers like you, like Archana ma'am, and like Virdi sir, you are updating the students, encouraging and inspiring the students. So it's my duty as the president of Microbiologists Society to appreciate you more such activities in the future. Thank you, madam. Thank you, sir. Thank you all.
Thank you, sir.
Thank you so much, sir, for your kind words. Now, I would like to request Dr. Archana ma'am, President, Delhi Unit MSI, to say a few words.
Thank you so much, Dr. Rashi. A very good afternoon to our most respected Professor Deshmukh sir and Professor Virdi sir, whom we know for the past 3 decades now. And always we have, as students, we have had the temptation to attend to whatever sir spoke ever. So that's why none of us miss any chance whenever we get an opportunity to hear to Professor Virdi. So, and also, I'm very happy. I congratulate Institute of Home Economics, Department of Microbiology, the coordinator of this program, Dr. Sunita, and also Dr. Sunita, my dear friend, and Dr. Aarti, who is the teacher in charge. We have grown together, so we are bosom friends. Also, Dr. Ashima, Dr. Nitika, and Dr. Sonia, and so many other are there. I was just watching. Dr. Manpreet is there, Dr. Neeru, Dr. Lakshna, Dr. S.K. Srivastav, and Dr. Nitin Chauhan. So the event is really star-studded, and what better way could there be of celebrating the International Microorganisms Day? So I'm very happy to be here. I congratulate the college, IIT, the institute, and we are very, very excited to hear to the challenges we face while dealing with the AMR issue. So we are looking forward, sir, for your talk, for a scintillating talk. Definitely, the problem, the menace of AMR can never be overemphasized, because this problem is actually a very big problem as compared to so many other challenges that we are dealing with, because of so many factors like indiscriminate use of antibiotics, etc., which sir will cover. And also a very good topic of Mighty Microbes by Professor Shilpi Sharma of IIT Delhi. We are very excited, and I congratulate, looking forward to the entire enlightening session. Thank you. Over to you, Dr. Rashi. Thank you.
Thank you so much, ma'am, for your kind words. Now we are beginning the webinar with our first speaker, Professor Js Vedi, sir, with his brief introduction. Professor Js Vedi, who served the Department of Microbiology, University of Delhi South Campus for more than 3 decades, is a well-recognized expert and mentor in the area of microbiology. He did his MSc Honours in Microbiology from Punjab University and PhD specializing in immunology from PGI Chandigarh. As a microbiologist, his major area of research has been the study of emerging infectious diseases, especially waterborne bacterial diseases and their public health relevance to India. His work on molecular epidemiology of Yersinia enterocolitica, an enteric pathogen, is well recognized as an original scientific contribution in infectious diseases. In recognition of this work, he was conferred the Y.S. Narayana Rao Award for Research in Microbiology by ICMR. Building on leads from the work on beta-lactamase genes in Yersinia and E. coli carried out in his laboratory, he is now engaged in developing a rapid point-of-care test for detection of antibiotic resistance using genomics and informatics approach in collaboration with bioinformaticians, clinical microbiologists, and microfluidics experts. This work was adjudged best and awarded the first Ideathon Prize by BIRAC. The team led by Professor Virdi further received funding as Discovery Award from Nesta, UK, and BIRAC for development of a prototype of this test. Professor Virdi was also awarded the Science Hero in Vitrogen Best Lab Mentor Award 2016 on a worldwide competition. As member of Association of Microbiologists of India, AMI, Professor Brady has served in various capacities, including its president in 2018 and 19. Now, I would like to request Professor Brady to please start the talk. Sir, can you please start? Thank you.
Thank you very much. Well, I would like to thank Professor A.M. Deshmukh, Dr. Archana, and Dr. Sunita Agarwal and their colleagues for inviting me for this talk on antimicrobial resistance. See, I'm very happy, and I regularly keep on seeing a lot of activities which are being carried out by Dr. A.M. Deshmukh, along with, all those students with whom I have interacted during my stay at South Campus, whether it is Archana or Dr. Sunita and their colleagues. So to start my presentation, here, can you see the slide?
Not yet.
Not yet, sir.
Not yet. Sorry.
सर नीचे जो एरो है ना सर एक,
हाँ, but I'm not able to see once I start presenting.
हाँ जी सर. सर आप इसको ना आप
Just hold on.
Yes, sir.
सर आप अपनी स्लाइड्स को कंट्रोल एस कीजिए, फिर वो प्रेजेंट होगा. कंट्रोल एस कीजिए, सेव हो जाएगा.
Just हाँ, किसको
सर कंट्रोल एस किया आपने सर?
Yes, Ashima, क्या करना है?
सर
See, once I start presenting, that share button is not highlighting.
सर आपने ऊपर से क्लिक कीजिए, जो विंडो है ना, विंडो में वो 3-4 विंडो आएगी, उसमें एक को आप क्लिक कीजिए सर, फिर वो शेयर बटन आ जाएगा सर.
Correct.
ऐसे आता है सर.
First you have to select the PPT.
हाँ जी सर
बाद
विंडो
में शेयर को कलर आ जाता है.
When you click the PPT, the share button will get activated, sir.
सर आपने पहले एरो बटन को प्रेस किया जो पॉइंटिंग अपवर्ड है.
हाँ, yes किया उसको.
फिर आपकी स्क्रीन पे बहुत सारी विंडोज आ रही होंगी सर.
हाँ, 3 आते हैं उसमें. Your entire screen, present your entire screen, a window or a tab.
तो आप एंटायर स्क्रीन भी कर सकते हैं सर.
हाँ, मैंने एंटायर स्क्रीन कर दिया.
नहीं सर, जो आपकी जो विंडो है ना, विंडो में भी आपको वो दिख रहे होंगे 3-4 वो जिसमें ये जो विंडो होती है ना सर, जिसमें भी आपको
विंडो में आपकी स्लाइड्स दिख रही है, आपको विंडो में आपकी स्लाइड्स दिख रही हैं, आपकी स्लाइड दिख रही है इसी विंडो में।
हाँ, स्लाइड्स दिख रही हैं, लेकिन शेयर बटन जो है ना वो हाईलाइट नहीं हो रहा।
सर आप स्लाइड को क्लिक कर दीजिए, उसके बाद वो एक एक्टिवेट हो जाएगा शेयर बटन।
होल्ड ऑन, ये
सर पहले स्लाइड्स को क्लिक करिए, फिर शेयर
सर
बटन।
विंडो में स्लाइड्स को क्लिक कीजिए, जो विंडो अपीयर हो रही है उसमें स्लाइड्स को क्लिक, उसको सेलेक्ट करने के बाद शेयर बटन हाईलाइट हो जाएगा।
ओके, जस्ट होल्ड ऑन। ओह या आई थिंक आई हैड नॉट क्लिक्ड दैट।
यस सर।
Okay.
Yes sir, it's visible now.
Okay. Is it now being seen?
Your screen is visible.
Yes sir, slides are visible.
Is it okay now?
Perfect sir, perfect.
Yes,
but let me see whether it is moving or not. Is it moving?
Yes sir.
Yes sir. Yes sir.
Yes sir.
Is that okay?
Yes sir, perfect sir.
Perfect sir.
All right, then I'll start. Thank you. See, in fact, antimicrobial resistance, you will see during my lecture, it is almost everything is a challenge there. While we start talking about addressing antimicrobial resistance, almost everything is a challenge. Just to introduce you to the subject, see, these antibiotics, they were introduced into the human medicine somewhere in the 1930s to treat bacterial infections. And these proved to be wonder drugs. These were responsible, not only that they proved wonder drugs for treating infections, these were responsible for an unprecedented advancement in human medicine. Why? Because with the introduction of these antibiotics, more and more complicated and sophisticated surgeries, they could now be performed. I mean, those surgeries which were earlier not possible, especially some of the complicated surgeries of orthopedic surgeries or brain surgeries, see, these could now be easily performed. Not easily, I mean, these could be performed and infections could be prevented. As time progressed, antibiotics started to be used for veterinary medicine, animal husbandry, agriculture, aquaculture, etc. See, the widespread use of the antibiotics led to widespread exposure of the microbes to antibiotics, and that led to emergence of antibiotic resistance. Why? Because these antibiotics, they kill microbes, and every living being on this planet has a survival instinct. Nobody wants to be decimated. Nobody wants to be killed or eliminated. So microbes started resisting the killing effect of the antibiotic, and that is how they became resistant. They became resistant. See, I'm not going into any of those mechanisms because microbes have got a variety of mechanisms. You name them and they have it for the development of antimicrobial resistance. They can hydrolyze the antibiotics. They can change their target sites. They can throw the antibiotics out and survive in the presence of antibiotics. So, but now here I'm going to introduce you to another term, that is AMR. The AMR is nothing but antibiotic resistance. The only thing is that this...
Excuse me, sir.
Yes.
Sorry to disturb you.
Sir, could you please hide this pop-up? There is a pop-up, meet google.com.
Okay.
Yeah, please. Thank you, sir.
All right. See, this, however, this term AMR is a more, you can say, recent and a more globally accepted term, antimicrobial resistance. Otherwise, it is nothing but antibiotic resistance, or what we have known as multiple drug resistance, multiple drug resistant organisms. But all over globe, this term is the one which is continuously currently being used to denote antibiotic resistance. Let's see why there is antibiotic resistance. One of the reasons for this is excessive use or misuse of antibiotics in human medicine. When I say excessive use or misuse, I mean that many times antibiotics, they are used where they are not required. For example, one of the very good example is the viral infections. And especially in respiratory cases, respiratory infections, it is very difficult without clinical test, very difficult to identify whether the infection is due to a bacterial infection or due to a viral infection. So antibiotics are used to treat some of these respiratory infections indiscriminately, whether they are of a viral origin or they are of the bacterial origin. When I say misuse, see, many times in the human medicine, there is a good misuse also. Why? At times when lower classes of the antibiotics may be warranted, the physicians, they tend to use broad spectrum antibiotics. Broad spectrum antibiotics, in fact, are meant to be used only for very serious infections or for very, you can say, serious surgeries, etc. However, when these antibiotics, these broad spectrum antibiotics, they are used for every other infection, the microbes tend to become resistant to them. So similarly, there is a use, or rather I would say misuse of antibiotics as food and feed supplements in the animal husbandry, only because they give a weight gain to the animals and are responsible for the profit to the industry. Otherwise, they are not doing any purpose there. Use or misuse of the antibiotics in agriculture, in aquaculture, there again, their use is associated with increased production of the produce rather than any treatment or any, you can say, judicious use for their infections. Of course, in aquaculture, they may be used for this purpose, but there again, at many times, these are used just to keep the fish healthy. Now, this is one story of where there is a use and misuse of antibiotics. But besides that, there are other problems. What are these problems? Residual antibiotics in hospital effluents are discharged in wastewaters. Those antibiotics, those residual antibiotics end up in wastewater. Then wastewater from pharma industry, manufacturing antibiotics, are discharged into wastewaters or rivers. When I say wastewater, I mean to say any kind of waste water bodies where polluted water is there. See, most of the rivers are also polluted, but and they receive wastewaters from urban areas. Runoff from animal farms, agriculture, they all end up in wastewater or rivers. So that means if you look at this story of these two stories of the exposure of the microbes to antibiotics, you can see microbes get exposed to antibiotics in two ways. Number one is the direct use of the antibiotics. That means when the antibiotics, they are directly used to treat human beings or to animals, or when they are given to food animals, or when they are directly being used as antibiotics. And the second is, wherever you use these antibiotics, they will finally end up in waste waters or in polluted waters or polluted rivers. They will end up where the microbes also end up. That means that these are the two places where the microbes are exposed to the antibiotics. And as these are exposed to antibiotics, they start developing resistance. As I said, that the microbes, they have got a large number of mechanisms by which they can undo the effect of the antibiotics, and that is what we call as AMR. Let's see what is the current situation. Now, a very large study in the United States in somewhere in 2014 by Jim O'Neill, it brought some very important insights about antibiotics. What is that? Number one of them is that as the amount of the antibiotic or amount of the antibiotic goes up, the resistance also goes up. That means the degree of the resistance or the quantum of the resistance of microbes is directly proportional to the amount of the antibiotics being used, the total amount of the antibiotic used. This is true for almost every country. So that means one thing is very important, that somehow for addressing the problem of the antibiotic resistance, we have to bring down the use of the antibiotics. If we are able to bring down the use of the antibiotics, it is quite possible that we have initiated a sort of addressing the antimicrobial resistance also. Some of the countries, especially in Scandinavian countries, where they very stringently restricted the use. I won't say they banned, because antibiotics, they cannot be banned. They have not been banned for, you can say, even environment purpose or even in animal husbandry, but they were very, you can say, strictly or stringently regulated there. They showed that the antibiotic resistance or antimicrobial resistance has come down in the subsequent years. So amount use, that somehow we have to restrict. So this is another current situation. This is what is projected, that the number of the deaths by antimicrobial resistance in 2050 will be highest as compared to any other disease. If we take cancer, which probably is one of the major killer, even the AMR will claim more lives than this. So that means we are currently in a situation where all these things are projected, but we still have the time. We still have the time to address it. That is why the physicians or all the stakeholders all over the globe, they don't want to be taken unawares, and they have started building up a sort of a movement all over the globe, so that this problem of antimicrobial resistance can be addressed from almost each and every angle. But nevertheless, addressing this problem is a challenge. What are those challenges? Let us see. See, this is another current situation. No antibiotics today, no cures tomorrow. That is what World Health Organization says. So this is how you can address AMR. See, develop new antibiotics. See, these are 1, 2, 3, 4, 5 different ways in which one can, each one of these measures can contribute to addressing the AMR. Develop new antibiotics, all right. Restrict or decrease the excessive use of the antibiotics, all right. Do not use antibiotics in animal husbandry, agriculture. But you will see each one of them has a challenge in it. Develop rapid methods for detection of AMR. It's a challenge itself. Develop new vaccines, because vaccines for infectious diseases, as the infectious diseases will come down, the use of the antibiotics will come down, and the AMR will come down. Develop rapid diagnostics for infectious diseases. So these are I would say that I have tried to put the things in a very succinct way. I have not. I could have listed so many things here, but then it will be more or less like a you can say a review article. No, we don't want that. We want something crystallized. Information should come out of it. So another thing which everybody or any stakeholder in this problem has realized that AMR is a complex problem. For example, if it is an environmental problem, it has agriculture also. If it is a human problem, then of course, as I said, human antibiotics, they end up always in wastewater, so it becomes an environmental problem. So similarly, in veterinary medicine, wherever you use in veterinary medicine or use in animal husbandry, they end up in the environment. So somehow it is now being suggested that 3 groups of the people, number 1, medicine people, the veterinarians, as well as the environmental people, they must interact more frequently to develop measures to tackle the AMR problem. And that is what is called as a One Health approach. In this, I just had a chance to see that there is, in Europe, they have formed a med-vet nexus, or med-vet. Med-vet means where the veterinarians and the medicine people, they interact more closely on subjects like AMR, as compared to what is currently being done. Let us see, let us look at some of these things one by one. Let's see. See, develop new antibiotics, not an easy task. Why? Because it takes a long time to develop antibiotics, almost 10 years. There's a high cost. Millions of dollars are required to develop even a single new antibiotics. Companies, they shy away from this. Some of the top, almost all top companies, they shy away from this because the profits are very low. And nowadays, these companies are more interested in the type of the diseases like diabetes or rheumatoid arthritis, where people will have to take their drugs throughout their life, rather than taking drugs like antibiotics, which are most of the time taken for 5 days, 10 days, or at the most, and are highly subsidized also. So nobody wants to develop these antibiotics. No new antibiotic scaffolds have developed in the last 3 decades. Can you imagine? No new antibiotics have been discovered. I said scaffolds. See, scaffold means the basic structure, the basic chemical structure of the antibiotics. Of course, we keep on developing new antibiotics by using, tinkering the scaffold molecules or introducing a variety of changes in them and getting new antibiotics with new spectrum, but we don't call them as new antibiotics. New antibiotics are only when you discover a new scaffold. The way we have got a entirely unique scaffold for the beta-lactams, we have a unique scaffold for the aminoglycosides, and so on and so on. However, with this kind of AMR problem being projected some time back, a group of 25 companies, they have given assurance to WHO that they would develop new antibiotics in view of the impending dangers. And WHO on its own part has given all kind of assurance that they will be given all kinds of tax benefits and other facilities to develop new antibiotics. Well, another thing is, you develop alternatives to antibiotics. Yes, fine. See, go for phage therapy. All right. You go for antimicrobial peptides. Yes. Antimicrobials from natural products. Well, there are other alternatives also. But remember, each one of them is beset with its own problems. Phage therapy, which was very common, not very common, but was practiced before antibiotics, they were in use. And in fact, phage therapy fell out of favor only because of the introduction of the antibiotics. Phage therapy has its own problems. Antimicrobial peptides, they were touted to be, you can say, very favorite in terms of their antimicrobial properties. But somehow, if we evaluate today, as we stand today, till date, nothing has been developed or discovered which rivals antibiotics. Remember, whether antimicrobial peptides, phage therapy, or other antimicrobial peptides, or this anti-quorum sensing agents, variety of things are there, but nothing has been developed or discovered which rivals antibiotics. So it is a real challenge, whether you are looking for new antibiotics or whether you are looking for alternative to antibiotics. We have to discover something which comes up comparable to antibiotics in terms of their effectivity and safety. Okay, but nothing is there right now. However, a very novel idea is that to develop antibiotics to which microbes will not develop resistance. Well, that is really a very novel idea. People have come up with some molecules, but then those molecules, they have not stood the, you can say, test of time. It's only time will tell how do we address this problem. Next, let's see. We say prevent the misuse of the antibiotics. All right, misuse of antibiotics in the human medicine can be prevented. How? Develop rigorous protocols for use of antibiotics. Yes, this is being addressed. The good hospitals, good institutes, they are all having very well developed hospital infection control mechanisms, and protocols are being developed when to use a particular antibiotic, so that the purpose is that you use only those antibiotics which can serve the purpose well and prevent the misuse of the broad spectrum antibiotics which can be preserved for more serious infections. Because if we keep on using these broad spectrum antibiotics indiscriminately, we will make them useless because microbes will develop resistance to them. Now, developing a rapid or a point of care diagnostic for AMR, it's a challenge. It's a big challenge. See, yeah, just before I forgot to tell you that these rigorous protocols are being developed. But what about the general physicians, those clinics where the physicians, they continue to use the broad spectrum antibiotics. So this is a big problem. But nevertheless, this challenge is slowly being met. Now, developing rapid point of care diagnostics for AMR or for infectious diseases, it's a challenge. Why? Because till date, whatever methods we have for knowing the antimicrobial resistance of the various offending microorganisms, they all take at least a minimum of 18 hours. Normally, they would take around 36 hours or so. And all over the globe, majority of the hospitals and clinics, they continue to take 36 hours, both for isolation of the microbes and then finding out the sensitivity. In some of the best institutions, probably because of the availability of certain instruments developed by certain companies like Biochemical or Abbott, that we have been able to bring down that time to 18 hours. But that 18 hours is too much. Once a person is detected to have an infection, you cannot delay antibiotic prescription for 18 hours. Some antibiotics has to be given, and it is during this time that the misuse takes place. So you have to develop an antimicrobial test which can be done on the bedside quickly. The results of the sensitivity are available, and the antibiotic can be prescribed immediately. Appropriate antibiotics can be prescribed immediately. So you can imagine how challenging it is to develop a test where you don't have to culture the microbes. I will tell you some of the approaches where this challenge is being met. Misuse of the antibiotics in animal husbandry or environment, stringent laws and regulations, they are still being developed. They are still being, you can say, being articulated all over the globe, because these kind of laws and regulations cannot be imposed in a jiffy. These things that do take time. And there is a challenge in that also. Almost major, you can say, regions of the world, countries, they are coming up with the appropriate laws and how to regulate them. Then misuse of antibiotics by public can be prevented by educating the public about hygiene, about the significance of the antibiotics, about AMR. But then that itself is not an easy job, because we have been trying to educate the public for almost 100 years about the hand hygiene and you can say other parameters of good health. But still we know that a large population of the world stays in unhygienic conditions. See, a situation like COVID has probably done a good job in terms of making people aware about hygiene, sanitizer, etc. But nonetheless, it is not. Nonetheless, it is not a. You can say is not enough. And then it has also brought the use of the antibiotics, high you can say amount of antibiotics use. So it's a, you can say, very you can say contradictory kind of a situation, where on one hand we say that the COVID has brought some kind of a advantage in terms of educating the people about hand hygiene and other things, but at the same time it has resulted into high misuse or high use of the antibiotics. So if you start analyzing each of these problems in that detail, you will see that there is a challenge everywhere. Significance of the antibiotics and AMR. See, making all these kind of things, for the lay public is not that easy. That's why you can see institutions like WHO, they keep on hammering, they keep on coming up with slogans, they keep on coming up with a variety of posters, educating programs, but it is a challenge, but it will again take time before we can you can say, bring that education to a level where it is going to make effective, is going to be effective. So let us see what else. See, as I said, point of care tests for microbiologists. See, this is the usual thing in a laboratory. See, the sample goes from the patient, the sample goes into the lab, comes back after after identification and ID back into this, or if it is an OPD, like this. So the time is lost. The time is lost here. That it takes approximately, as I said, 36 hours or so. But the patient here cannot be left with antibiotics. Here in outpatient departments, nobody does any kind of antibiotic testing, or you can say what kind of infection it is. The patient comes and the antibiotics are prescribed. And the catch here is that it is always a broad spectrum antibiotics which is prescribed here, and it is maybe prescribed even in a situation where it is not required in viral infection. So there is both excessive use or and a misuse of the type of the antibiotics. So that means these are, see, this is the usual you can say train of the events. What happens when a person has an infection and when it goes to a doctor. Now, this is without any kind of a test for antibiotic, whether what kind of an organism it is, what kind of antibiotic resistance it has, and what kind of an antibiotic is required. It is done without that, and there is the danger, or not danger. I would say rather this is rather widespread. This is widespread all over the globe, where this is responsible for excessive use of the antibiotics as well as the use of the broad spectrum antibiotics, and there you can say they're getting nullified in the process because of the resistance. Now, these are the tests which are available for finding out the organism we have, as well as what is its sensitivity, and trying to find out the best antibiotic. But each one of these takes a very long time. This is the Kirby-Bauer method. This is the dilution method. This is the latest e-test, which is now replacing all these things, and you can directly find out the MIC from this e-test. So that means if we have to replace all these things, we must replace it somewhere here. But if you want to replace it, it has to be rapid test. Okay, so rapid test, as I said, these are the rapid test. But as far as the current development of the rapid test is concerned, you can see the people have come up some imaging technologies. See, something about a year or a half back, this kind of a imaging technology was not even thought of for antibiotic resistance, because we always thought that we have to culture microbes, and without culturing we cannot determine its sensitivity or its resistance to antibiotics, where it is only this challenge of the antimicrobial resistance in the last 4 to 5 years that people have come up with totally novel technologies and imaging-based technologies, where you simply, and I'm not going into the details of this, where you expose your bacterial cells only for a couple of minutes, and then take those bacteria, study them under a microscope, millions of those cells under a microscope, and try to find out whether the microbe is resistant or it is sensitive. That means by simply looking at the morphology of the microbes, how the morphology is changing after exposure to a variety of antibiotics, in a couple of minutes, you will be able to find out whether they are resistant to a particular antibiotic or to or they are sensitive, as a result of which a you can say, a very appropriate antibiotics can be prescribed with a minimum period of time, maybe less than within an hour, within 1 hour. And this is but this test is still far away from a point of care format. This proof of the principle for this has been demonstrated, but it may take still more time, a couple of few more years before this kind of a test is available for real use. Next one, MALDI-TOF. MALDI-TOF is something which is used for for protein analysis. See, a few years back when MALDI-TOF, it was unheard of that we will use it for antimicrobial resistance or we will use it for bacterial identification, but now it is being used in big hospitals for at least for identification of the bacteria. It is being used for antimicrobial susceptibility testing as a rapid test. MALDI-TOF is being you can say is being adapted or is being studied, because after all this machine has to be miniaturized and before it can be really used, it can be really useful as a point of care test for detection of AMR. So it is in this line only. See, when approximately 4 or 5 years back, when there was a lot of you can say a lot of concern about the AMR and lot of funding agencies almost all over the globe, they were calling scientists for developing antimicrobial tests. That in our own country, some of DBT and BIRAC from the UK, which is a charity in UK, they called scientists to develop this kind of a test, and this it is at that time that our group with the students and you can say collaborators from Department of Biophysics and some from IIT and from Safdarjung Hospital and my lab, that we put together certain you can say ideas that for the rapid development, and this is how we got funding, not only the immediate funding but also later funding in another competition in UK, and that is what we are now in the process of development, but that's a story for another time. See, I would like to thank all these students who have been at one time, to start with, many of them, they studied the Yersinia, and slowly, because Yersinia has got a lot of antibiotic resistance, especially of the beta-lactam type. We started beta-lactamase type. We started studying these beta-lactamases and slowly got into AMR and finally into the development of the AMR and development of a rapid test for AMR. And with this, one of the most important thing I would like to say is all this knowledge, all these students, their knowledge, their learning, everything has been possible with the generous funds from these people. Without that, nothing would have been possible. And with this, I would like to thank you all for your patient hearing. And if you have any questions, I would like to interact with you. Thank you very much.
Thank you very much, sir, for your nice presentation. The floor is open for questions now.
Can I ask one question, sir?
Yes.
Like these days, there is a concept that even these antibiotic resistant genes are present in the environment.
Yes.
So, sir, if we are considering these tests for the patients, so what will we do about these genes which are present in the environment? How can we tackle that?
No, can you make the question more clearer?
Sir, I have listened somewhere that antibiotic resistant genes are present in the environment.
Yes.
So how can we tackle these genes if they are present in the environment?
Yeah, you see, in fact, if you ask me, there is no way how you can tackle that. See, that's a big problem. That's a challenge. There is no way you can, tackle them. They will remain there. However, if we have the knowledge of those genes, we would at least have an idea what kind of a resistance is going to emerge in the near future. Okay, because there may be a type of a resistance which is not prevalent in our country, but it is around the corner. It is somewhere in another part of the world, and there may be some kind of signs and indications that you might also be having this kind of a resistance in future. This is how you address. Or maybe if you know in a particular geographic area, for example, let's say North India, what is the type of the genes present in the environment, the antibiotic resistant genes present in the environment. From here, what kind of strategies for the treatment of the animals, treatment of the human beings, they are to be carried out. That can be, some inferences can be derived from that. Okay. Similarly, the type of the resistance which is there in the south, for example, what kind of treatment strategies, what should be used, what should not be used, what kind of already resistance genes are there in the environment. A lot of these strategies can be derived. But if you ask me how to address those resistance genes, we have nothing. Sometime back, the DBT has asked that if anyone, or BIRAC had asked that if anyone has got some ideas for addressing the presence of the AMR, antimicrobial resistance genes in the environment, come up. But then there was nobody. I was there in the, you can say, as a part of that, you can say, giving those projects, but no one came up with this kind of a proposal.
Thank you, sir. So it means if we know the genes which are prevalent in particular area, we can modify our treatment strategy.
Yes, treatment and our not only treatment. See, variety of things. Whether these kind of antibiotics, they are to be used for animal or veterinary medicine or not. Those kind of things. Human, fine. Other things also can be used. Or looking at these genes, you can also tell whether these are the genes which will rapidly transmit to other parts of the country, whether there is a colistin resistance is there, for example. If we really do not know whether colistin resistance is there or not, we will be totally, you can say, unaware of that. But once we know that whether this kind of a resistance will spread in our country fast or it will not spread it. So as a result of which you devise your strategies accordingly. You become aware.
Thank you, sir. Very nice lecture, sir.
Yeah.
So, can I ask something?
Yes.
So you said now instead of culturing the bacteria,
yes,
we can expose the bacteria to antibiotic for some time and then note the change in its morphology.
Yes.
So, and can you please illustrate more on this? Is this some new technique, and how many, is there some standardized protocol in how much time you check the morphology?
Yes. This is not a new technique. This is a breakthrough in microbiology. If you ask me as a microbiologist, I would say it's a breakthrough in microbiology. See, nobody ever thought of morphology, studying morphology of the bacteria and trying to find out the resistance. Okay,
right.
Slight change in the morphology. Of course, as I said, this is a complicated kind of a thing that if you expose bacteria to any antibiotic for a couple of minutes, it will change its shape, but it will not give you any idea. So of its sensitivity. But you have to study or you have to image thousands or hundreds thousands of those cells. That means you should have such fast imaging where you hundreds and thousands of the images of the bacteria can be taken and then they can be analyzed, where again that analysis is primarily a you can say artificial type of an intelligence, where you put each and put several dots on each and everything. See, you must have seen the way the faces of the people, they are constructed using the artificial intelligence. This is how you construct bacteria, the bacterial morphologies, and find out, pulling the data from millions of such cells, you find out some authentic differences in the cells following their exposure to antibiotics, and then trying to find out whether it is this morphology change represents resistance or it represents sensitivity. It's a big...
Right. So you're trying to say this is a more efficient and faster method as compared to culturing the conventional methods of culturing the bacteria.
Yes. See, culturing means 18 hours, minimum of 18 hours. Under the best laboratory circumstances, 18 hours. But this will be 1 hour, 1 hour.
And this may be true for non-culturable bacteria also, no? Yeah.
Yes, non-culture, but as long as we can see their morphology, some exposure or some growth, no, but I think some growth should be there. Non-culturable, if they are not growing, no change in morphology after their exposure to antibiotic, it will be difficult to apply it to non-culturable bacteria.
True. Thank you.
These developments are only, these imaging technologies for AMR, it is only a development in the last 2 years, I will say, less than 2 years. Less than.
So you can share the reference of these, please.
Oh yes, nice. Many.
Please share the reference. And sir, this Malditoff also that you talked about, if you can share the reference.
Many of them. I will give you, share you.
Thank you so much, sir. It was an excellent talk. Thank you.
Thank you.
I want to ask in continuation, sir.
Yes.
Yeah. Sir, is it necessary that the morphology change has to be there? I thought it may or may not.
No. Without morphology change, what you will study with the imaging? At least some minimum change should be there.
Sir, so we can say that this imaging-based technology is true for those drugs which perturb the morphology of the cells.
Yes.
This is only for those drugs. No, otherwise, because just imaging, we cannot go
exactly
into the cell and look for the DNA or some molecular base.
Yes. Okay. Those drugs which perturb, those antibiotics which perturbs the morphology.
Just morphology, cell wall, or just like. So we can opt this method for those things only, not for every. It is not true for every mean drug.
Yes. So you can still see that final verdict still is not out. We really don't know what will happen. The company says that they will be able to make a test in another 3 years or so.
Yes, sir.
Okay. Yeah. Then since you have brought this point of morphology, point of morphology, that change in morphology, remember things like flow cytometry. People now they have come up with very innovative ideas that they are trying to use expose antibiotics, and instead of morphology, they are simply looking for changes in DNA, changes in mRNA. So where flow cytometry is also being used for antimicrobial testing.
Yes, I got your point, sir.
Yeah, but then again, you see, the big point still remains, because flow cytometry has to be adapted to a point of care also.
Mm-hmm.
See, similarly, Malditoff is a big machine. Otherwise, if you have to do you can say AMR, antimicrobial testing, in a big hospital, I'm sure within 2 years we will be doing our antimicrobial testing with a Malditoff. Okay, but that will be possible only in big hospitals. That will address our AMR in a very minuscule manner. What we need is a kind of AMR test or a point of care test or a rapid test where it is available to all physicians, to all, you can say our, all these doctors who are sitting in our localities and all nursing homes where it can be quickly performed. Then it will make a real change in the use of the antibiotics, or it will bring down the use of the antibiotics. So you can see how big the problems are. It's a challenge.
Yes, sir. True, sir. Thank you, sir.
So, can I ask a question, please?
Yes.
Very brilliant talk, sir. Nice to hear you. It's always a pleasure to hear you and get educated. Sir, my questions, there are two questions, quick questions. One is why phage therapy is not being promoted in India. Because in the erstwhile Soviet Union and the Georgia and other states, it's a very big industry, and now the US is also investing into it because of late many patients in the United States who were the bacteria were resistant to all the antibiotics available, and their lives have been saved by the use of phage therapy. So why India is so much behind in this? And
the main thing as far as the phage therapy is concerned is, after all, you are going to use a living microbe or a phage for a human being. See, some kind of a committee and some kind of very, you can say, chivalrous people, they should come up and they should develop those guidelines and say that we need it in our country. We must have it. There is nobody, in my opinion, nobody has come up with that kind of a confidence. We must introduce it to our country. That is probably the lack of it. Who will bell the cat? A kind of a person, the chairman who is a member of the committee, and they are going to introduce phages. See, somebody should have that courage. I think that is needed.
But the competence in our country is there. I mean,
no, I don't think. As far as phage therapy is concerned, competence is not there. See, as far as these Poland or other these East European countries you are talking about, see, these people, they were practicing, even in Russia, they were practicing phage therapy before the introduction of the antibiotics in a big way. And look at the way they are isolating their phages, they are characterizing them and keeping them. You will be surprised. Our you can say expertise, although we have the infrastructure and other things, but our expertise has not reached that level.
And the other thing I wanted to ask was, India is far behind in putting these bans on the sale counter, antibiotics. So that also needs to be addressed as a policy matter.
See, Sunil, you look at this. See, ours is a LMIC. See, low and middle income countries, LMICs, lower and middle income. See, where antibiotics are playing a very important role. See, you cannot ban them quickly. Where you will get them? Where infection rate is high, we cannot totally ban them. Sometime back, a lot of restrictions they were put. Red line was put on the wrappers that these will not be antibiotics. Wrappers will not be sold over the counter. See, but ours is not ours. Most of the developing countries, it is very difficult to ban, kind of a ban or regulate, because after all, these free use of the antibiotics doing a lot of purpose. See,
but we are facing a lot of criticism from the West that India is very much responsible for many of the antibiotic resistance that has come about. So, we have to have a balance in our country to solve this problem, because the Netherlands and the Scandinavian countries, they have shown really a good model to
lot of regulations are being put. I know a lot of documents have been prepared, a lot of these instructions they were issued, and some measures were taken also, or they are being taken. But as I said, these are a challenge. You may say you ban the over-the-counter use of antibiotics, but this is not possible in a developing country, or in a small country, you can say developing country. It may be possible in a highly developed country where everyone is so well off that they can immediately go to a physician, pay his fees also, get the test done also, pay that also, and then buy the antibiotic also. But in our country, that's not possible.
Right. Okay, so thank you so much. Thank you.
Thank you.
Thank you so much, sir, for this wonderful talk. We have a few more questions in the chat box. So do we have time to address these?
Just let me look at.
Okay, so there's a question from a student. Sir, during COVID times, we are dealing with a virus. Then why are doctors prescribing antibiotics?
Doctors are prescribing these antibiotics because we know it is virus, but these are antibiotics that are being prescribed to those patients who suffer from secondary infections. Once they have COVID infection, they are having other infections also. So antibiotics are being prescribed for those infections, not for the COVID. Not for the COVID.
Sir, can you tell us some AMR bacteria that have been developed because of the indiscriminate use of broad-spectrum antibiotics?
Almost everyone. Klebsiella, Acinetobacter baumannii, E. coli, all of them, they are because of the indiscriminate use.
Pseudomonas aeruginosa.
Pseudomonas aeruginosa, yes.
Staph aureus. Staph aureus is also.
Staph aureus, yes, you name it. The escape organism. See, we call them as escape organism. Okay, E for E. coli, S for Staphylococcus, K for Klebsiella, escape organisms. They are high priority organisms. Except maybe I think Pseudomonas aeruginosa, because I won't, Pseudomonas, yes, because Pseudomonas aeruginosa has inherent resistance also. But then besides inherent, it has got added also lot of resistance to it, which is because of the misuse. So yes, everything is due to misuse.
So we have one more question from Archana.
So you see, you take the case of Staph aureus. When in 1920s, when the penicillin was introduced, this was a wonder drug for Staphylococcus aureus. Staphylococcus aureus, it was a wonder drug. It will take, you can say, decimate the Staphylococcus aureus within seconds or minutes. It will kill the bacteria, and no trace will be left of the Staphylococcus aureus. And today, Staphylococcus aureus, it is all resistant to penicillins and all that.
Sir, MDR.
Sir, actually, Alexander Fleming saw it on the lawn of Staph aureus only.
To
start with, yes.
Yes, with the Staph aureus only, yes. And today every staph is resistant to penicillins. It is their exposure only, and excessive exposure. If we are able to limit that exposure, probably we would not have that kind of a situation. And this warning was given by Alexander Fleming himself. And it is only in the last few years we have dug up those warnings given by Alexander Fleming. In the last 60 or 80 years, we have never talked about what kind of a warning was given by Alexander Fleming. It is only when the AMR has spiraled into this kind of a problem that we have dug up and found out that he said this kind of a thing.
So it's like children, when they grow up, they understand what the parents were saying 20 years back. It's similar to that.
Yes.
So the next one is my question quickly, because I think the next speaker, the time is due. Sir, I wanted to know, Dr. Rashi, may I ask?
Yes, ma'am. Sure, ma'am.
Yeah. So, sir, are there already any guidelines and newer rules for the restriction of use of antibiotics in food processing industries and all, in agriculture, and also Similar thing in the hospital waste management, sir.
Yes. Already they are being developed. That they have rather been developed, and the nodal agency for this is that Center for Environment. That Sunita Agarwal or what? That down to earth.
Sunita Narayan.
Sunita Narayan. Yeah.
That is the nodal agency who are developing all these guidelines. Okay, ICMR for medical people, it is ICMR. Each one have formed a very big group where they have taken a variety of people from different disciplines to those guidelines. And remember, these guidelines are being developed not only in our own country, they are being developed almost all over the globe. It has been made mandatory by WHO. Okay, it has been made mandatory by WHO that these kind of guidelines should be ready by this and this date by every country. See,
sir, what about the stringency of following these in actual?
People question that. They say that you may lay down the rules, but who will follow them, or who will make sure that these rules are being followed? Regulation, you mean? Okay, that is also being discussed. That you should have a regulatory agencies also who would properly check that the rules are being followed or not. Appropriate penalties being you can say levied for them, all those kind of things.
Thank you so much, sir. It was very much eye-opening and enlightening, sir. Thank you, Ayuchi. Thank you, Dr. Rashi.
My pleasure. Thank you very much.
Thank you, ma'am. Thank you so much, sir, for this wonderful talk. Now, I would like to request Dr. Aarti Nigam, our teacher in charge, to give vote of thanks to sir.
Hello, everyone. Today we are celebrating International Microorganism Day. It has been our pleasure to have Professor Deshmukh, Professor Virdi, and Professor Shilpi Sharma for our program. These experts are well known in the field of microbiology, and their presence in our program will surely motivate faculty and students. On behalf of entire faculty and students of Microbiology IHE, I wish to express our gratitude to Professor Virdi for accepting our invitation for this talk and for delivering such an informative talk. The faculty members have been attending your lectures since long. Now this batch of students has listened to you first time, sir. I sincerely hope that they are thoroughly benefited from this talk. By such a well-learned speaker and expert such as yourself, as well as from the relevant theme around which you spoke. The problem of AMR and strategies to combat AMR are important area of research, and all these aspects you have covered in your talk. We express our deepest gratitude to you, sir, and hope to invite you in near future, once our college reopen, to give talk to our student. Thank you, sir.
Thank you, Aarti. Thank you very much.